So another thing I need to mention is that we will have 5 minutes of Q&A after each talk. If you have a question for one of our speakers, then please raise your hand using the buttons indicated here, or drop a line in the chat room, and we'll see if we can answer that question during the 5 minutes. So today this seminar is called Tracing Trade, Crossing the Border Between Taxonomy and Forensics Against Illegal Plant Trade. This seminar is officially hosted by KEW Gardens, but also supported by the Gothenburg Global Biodiversity Center in Sweden and the Systematics Association in the UK. And there's actually quite a good reason for KEW hosting this seminar today, because at KEW Gardens we have quite a long history of plant identification. So as you all probably know, KEW Gardens has outstanding plant and fungal collections, mostly known from the herbarium and the living plants that we have in our gardens. But we also have a very exciting economic botany collection, which is basically a collection of mostly historical specimens linked to documenting the use and trade of plants by humans through time. And already several decades ago, KEW scientists started documenting all these artifacts and creating reference databases with information for important specimens in the trade. And this information is now being translated into molecular identification approaches, so using DNA sequencing. In fact, KEW Gardens was at the forefront of pushing for a unified DNA barcode for land plants, most famous from this 2007 paper by Mark Chase and colleagues from the Jodrell laboratory. But unfortunately, these traditional DNA barcodes are not useful in every plant group. Some plant groups are a bit trickier because of high diversification rates and other processes happening. So we're moving towards next generation sequencing approaches. And a good example at KEW of that is a recent collaboration between palm taxonomists and phylogeneticists with IKEA, where they try to identify the rattan palm species that are used for the canes in their furniture. And that is to improve the sustainability of the trade internationally. But KEW Gardens doesn't only work on plants. We also have a very large dedicated mycology team who are also using DNA barcodes for molecular identification. And there have been some very exciting examples recently. For example, some scientists from KEW did a study on a porcini packet from a London supermarket, in which they sequenced 15 mushrooms, among which they found 3 new species in the same packet that haven't been described previously. And more recently, there has been collaboration with the Millennium Seed Bank, which is a large conservation effort to bank all the seeds of the plants in the world. And they've sequenced the fungi living in the seeds from the Millennium Seed Bank and discovered countless new species to science. KEW also hosted a Plant.ID project, which was my PhD, supervised by Olwen Grace. And we work on the genus Aloe, which is a CITES restricted plant group, so international trade is limited. But as I said, these traditional DNA barcodes don't work in every plant group, and this is also the case for Aloe. So we've used the next generation sequencing approach to do DNA barcoding with some 200 nuclear genes. And we've actually managed to get our hands on some samples that were intercepted by border control at London Heathrow Airport. And we've identified them with this new tool to the correct species. But we won't talk about that much today. We have other people speaking. So these are the 15 students in the Plant.ID cohort. And today we will hear from 2 of them, from Mehrdad and Maurizio, who actually both work on illegal timber trade. This is the schedule for today. So these times are in Central European time. So if you're in the UK, please deduct 1 hour from this schedule. I've just given you the introduction, and then we will have the first two speakers, after which we will have a 15-minute break. And after the break, Olwen will take over from me from chairing the rest of the session. And we should be finished around 4 p.m. UK time. So then let me go to our first speaker, which is Siri Abihudi. And we are very proud to have an African ethnobotanist on our panel today. Siri is from Uganda, where she studied environmental sciences at the Makerere University. But nowadays she's in Tanzania, where she did a master's in traditional medicine development at Muhimbili University. And she recently completed her PhD thesis at the Nelson Mandela African Institute for Science and Technology, where she focused on the ethnobotany of the genus Aloe in Tanzania. She's currently back at Muhimbili University to focus more on plant propagation techniques that can help with plant taxonomy, ethnobotany and conservation. But today she will talk about her PhD thesis. This talk is pre-recorded, so I will present the presentation to you. But afterwards, Siri will join live for the questions to answer any questions you may have. So please give a warm welcome to Siri and enjoy the talk. And I will just have to unshare my screen and share the other one. Let me see. Of course, now I can't find it anymore. Sorry, just a second. Here we go. Let me see.
Hello.
Right.
I am Siri Abihudi, a PhD student at the Nelson Mandela African Institute of Science and Technology. And I'm here to present one of my objectives in my PhD dissertation titled Ethnobotany of Traded Aloe Species in Tanzania. I'm supervised by Professor Anna, Professor Hugo, and Professor Mahuna. Aloe species are found in mainland Africa in tropical and subtropical latitudes. And these aloe species are found in extensive altitude range. Some of these species, like Aloe boscaweni, are found along the coasts, but others are found along the high mountains. Like this one, Aloe lactosiphone, is found along the slope of Mount Usambara. But also we have this other Aloe volcaceae, which is found along the slope of Mount Kilimanjaro. In Tanzania, we have a total of 52 aloe species. Among those, 25 are endemic, 13 are threatened, and 5 are critically endangered. Use and biocultural value of these aloe species are not well documented. There is only one study on the ethnobotany of aloe species in few villages in Morogoro, Mbozi, and Kigoma that was done by Amir. And morphological taxonomy of aloe species is problematic, as most of these aloe species look alike. We have these two tall aloe species, Aloe Bali and Aloe volcaceae. They look so alike and differentiated by the locals. Aloe species are locally endangered, and the main threat to these species are anthropogenic activities and climate change. And there is no study on aloe species trade in Tanzania, so there is need to determine which species are highly threatened. Which species are highly traded and which regions are product in the material method for objective 1, 2, and 3. Regions with high diversity of alloy species were selected, but also included the Serengeti district, which has never been surveyed before. So we surveyed the regions of Mara, Kilimanjaro, Tanga, Katavi, and Rukwa. We did purposeful sampling to select these 5 regions, but also to select the districts. We skipped the districts where urban areas are found, like Moshi. Here in Kilimanjaro, we skipped Moshi. In Tanga, we skipped the Tanga urban. The same with Katavi. Sampling technique. We did random selection of respondents. These respondents were given consent form before the interview. We did semi-structured questionnaire to collect the information from the respondents and field assistance to our local people who are familiar with the geographical location of the area, but also familiar with the language of a particular area. A total of 237 respondents were interviewed, 21 from Tanga, 22 in Mara, 56 in Kilimanjaro, and 67 from Katavi. Analysis, we did Whitney U test to test age group and ethnobotanical knowledge if there is any correlation, but also to test age group and perception on the possible disappearance of alloy. And we did Kuskoalis tests to see the correlation between perception on the possible disappearance of these alloy species and ethnobotanical knowledge. Frequency and percentage were determined and photo were taken. For objective 3 and 4, we selected cities where traded alloy products were collected, and these cities include Mwanza, Moshi, Arusha, Tanga, Dar es Salaam, and Dodoma. Result for objective 1 and 2, full taxonomy and flowering time of alloy species. Characterization of alloy species by locals followed the morphological features, uses, and where they were found. Most of these species were identified based on their morphology, like a tall alloy, a short alloy, red alloy, spotted alloy, and for some people, their identification based on the use, like alloy for beer brewing. That's how they identified the alloy. But for some, they used the location where these alloy species are found, like alloy of the rock areas. These are the alloy that were only found along the rocks area. Morphological similar species were considered to be the same. As I said before, alloy volcansea and alloy bali were considered to be the same fish by the locals, and alloy rabaensis and alloy pavidensis were perceived also to be the same. And most of the local members of the community perceived all the alloy to be aloe vera when they were using the Swahili or English name, but when they were using the local, that's how you can differentiate between one alloy and another. The flowering time of alloy species followed the rain season, and in Tanzania we have 2 rain seasons, the short one, which range between November, December there, where most of the aloe in Serengeti district are flowering, and the rest of the aloe in Kilimanjaro, Tanga, and Rukwa, they were flowering during the long rain season, which is in February to early May. Ethnobotany, 20 useful alloy species were identified among the 23 alloy species, and alloy lateritiria, duperia, and saboritora had the highest number of uses compared to other alloy species. But we're surprised to find alloy leptosiphone and alloy flexilifolia, which are the critically endangered alloy species, to be among the highly utilized alloy species in their regions. These are some of the most used aloe species. This is Aloe lateritiaria, Aloe duqueiri, and Aloe secundiflora. Among the 23 aloe species, 3 of them were not utilized, and this include Aloe confusa, Aloe fibrosa, and Aloe boscaweni. The Aloe confusa was not even known by the tour guides along the Lake Chala, who have been there for almost 10 years. So it was not known by any, and we were the first one to introduce it to some of the tour guides along the lake. And for Aloe boscaweni, it was only known by the fishermen and the people who work along the coast. And leaves were the most utilized part of the plant, followed by the stem and roots. And soaking was the main method of preparing this aloe for medication, followed by boiling and gel. Most of these aloe species were used for treating malaria, Newcastle disease in chicken, stomachache, wound, and for making local beer. And these were mostly for the leaves, but for the stem, they were mostly used for making local beer, and others were using it for stomachache and female infertility issues. And aloe barley, followed by aloe volcaceae, aloe secundiflora, and aloe rabaensis were the mostly uprooted aloe species for the uses I mentioned. And these were the most uprooted, as I mentioned, aloe barley and aloe volcaceae. You can see they look different if you look closer. And the main administration method was through injection, followed by application on the skin and hair. And some of the aloe species were reported to be poisonous. As we know, aloe barley has been reported by Newton to be poisonous, but 2 respondents mentioned aloe secundiflora to be deadly. Effectiveness and side effects, 97% of the respondents said aloe is very successful in treating their ailment, and only 3% mentioned the aloe to give only temporary relief for some condition like diabetes, toothache, and numbness. Side effects of the aloe species include diarrhea and vomiting. Community perception on the disappearance and conservation of aloe species. So most of these aloe species were sourced from the wild, as you can see from the graph, and only few were highly cultivated, including aloe masawana in Tanga region and aloe noti in Katavi. And leaves were the main section of the plant that were highly utilized, except for some few species like aloe barley. And most of these aloe species were perceived by majority of members of the community to be threatened, as you can see from the chart. And few were considered to be still available, including aloe flexifolia, aloe secundiflora, and aloe volcaceae. Decline and conservation of aloe species. Some of aloe species were considered to be locally extinct by the respondents, including aloe barley in Kisiwani and aloe volcaceae in Kideleku. Age did not influence perception of the decline of the aloe species, and the number of users did not influence reduction of aloe species. In the last 5 years, majority of respondents have observed the decline in the population of these aloe species. By factor for the decline of aloe species, most of these aloe species were highly affected by human activities, including fire, clearing the agricultural land using fire for Aloe lateritiae and Aloe dorothei, but wrong harvesting method for Aloe secundiflora in Same district affected Aloe secundiflora, but also harvesting in bulk for trade was also a threat for Aloe secundiflora in Same district. For Aloe leptosiphone, it was highly affected by agricultural activities. People are planting maize close to their habitat. And for some few species like Aloe duquerie was affected by these insects. And for Aloe lateritiae in Tanga, it was highly affected by monkeys. They were eating the leaves and the fruits. And for Aloe volcanic were highly affected by elephants as they were close to the national park. Some of the medicinal plants were also highly affected by overharvesting for medicinal uses, and for aloe in Tanga regions were affected by the kids who were overharvesting the aloe for gel, and this gel were using as a lubricant for their local bicycle called Kikwata. And for Aloe boscaweni in Tanga, it was highly affected. It was assumed to be highly affected by the salt ponds along the oceans, but also it has been mentioned by the IUCN Red List to be affected by the salt harvest in Tanga. Local trade for aloe species, Aloe secundiflora was highly traded in Same district and has been mentioned to be affecting the aloe in Same. Aloe barley in Kisiwani was assumed to be extinct, and Aloe volcanic in Kidelepo was also assumed to be extinct, and this is because of using these two aloe for beer brewing in both areas. Aloe flexifolia was also assumed to be declining because of bulk harvesting for trade in Kilimanjaro, in Moshi and Arusha, but also in Dar es Salaam for making aloe products. But the price for the aloe leaf were very small, like it ranged from US dollar 0.04 up to 0.43. So it was very little amount to buy the leaf. Trade, these were the aloe that were affected by trade, the aloe secundiflora and stem of aloe barley was highly sourced for local beer brewing, which was for objectives 3 and 4, aloe trade in urban area. A total of 46 aloe products were collected. 13 were used for skincare products, and 33 were for herbal medicine. Skincare products include soap, shampoo, hair lotion, and body cream. Majority of the aloe products were well labeled, as you can see from the photo, except for few aloe species that were locally sourced. Products were sold outside the Moscow in most areas. They just put a table and put their product. And in some few shops in Tanga, at least they had a shop. They were selling inside the shop. And also in the Dar es Salaam local markets for herbal products in Kariakoo, they also had shops for these aloe products. Company for skincare products, most of these aloe were aloe products for skin care were produced by Tanga Pharmaceutical and Plastic Limited, followed by Grace Product Limited. And these were produced in Tanga in Dar es Salaam, but the market was observed to be high in Mwanza, Tanga, and Dar es Salaam. So Mwanza was not the producer, but was the only region that sourced most of the products from Tanga and Dar es Salaam. State of the product, most of these aloe products were in liquid form, as you can see. Others were in soap, like solid state, liquid, and powder. And these aloe products were highly administered through injection, followed by ointment and washing. Trade in aloe products. Aloe vera is a common name for all the products that were in the market, and the mostly widely distributed products include Samarati, that was found in Mwanza, Moshi, Arusha, and Dodoma. Vera syrup was found in Arusha, Mwanza, and Moshi, and the rest were also highly traded products. Regions with high diversity of aloe products. Similar products in different regions. Similar products were found in different regions except on the powder that were locally sourced. And Mwanza was the main city with high number of aloe product diversity compared to other regions. Use of the aloe products were mainly used for sexually transmitted disease, irregular and painful menstrual periods, swollen and painful legs, hernia, typhoid, and malaria. If you compare the cities and the rural area, the urban area had different uses of these aloe products compared to the areas. In my conclusion, genus aloe has multiple uses, as we have seen. We need to do more on the pharmacological and nutritional studies on aloe, but also aloe species in Tanzania are declining due to human activities, and there is need to continually update the species conservation status as it keeps changing with time. Implementation of laws and policies to protect the natural resources from anthropogenic activities are highly needed to protect these species, especially the rare species which are highly traded or highly threatened in their restricted geographical range. Ex situ conservation is also important for the traded aloe, so we can reduce pressure on the wild habitats. Thank you for listening.
Right. Thank you, Siri, for this excellent presentation. Let's see if Siri is here. Are you around, Siri?
Hello, I'm around.
Hey, welcome. Thanks for joining.
Thank you.
Was a very interesting presentation. I didn't know that aloe was used for beer brewing, for example, so that's quite cool. Does anyone have any questions for Siri based on the presentation? Please raise your hand or put something in the chat if you feel more comfortable with that. Otherwise, I can start with a question. Oh, Olwen has a question. Yes. Please.
Hi, Siri. Thanks so much for a fascinating talk.
Thank you.
I was very interested to see that there was only one species that you recorded that was used for its stem. And I wondered if you could tell us a little bit more about that.
I think it was aloe barley, where they used the stem for beer brewing, as I said before. Yeah.
That's interesting. And then if I can just follow up with my main question, you note that the most commonly used vernacular name increasingly seems to be aloe vera, which of course is the Latin name of a different species of aloe, the one that is most widely used internationally. So do you think that there is an opportunity to expand the cultivation and trade in aloes in Tanzania as a replacement for aloe vera?
I think there is need to do that. And I think we have some few farms for aloe in Moshi, though it's not aloe vera. So people are trying to cultivate some of these aloe, which are native to Tanzania.
Mm-hmm.
Yeah.
Thank you.
Thanks.
Yeah.
There is a question in the chat room, actually, Siri. I can read it out for you if you want. David Newton asks, are any aloes used for the extraction of bitters, as is done in South Africa with aloe verox?
Okay.
Sorry, what did you say?
We have this aloe secundiflora.
Mm-hmm.
They've been using its, can I say gel? I don't know it's for what, but they have been extracting the gel from aloe secundiflora. Yeah.
Okay. But for aloe verox, what they use is the colored leaf exudate. Are there any species in Tanzania that are being used for that?
No.
Okay. Okay, that's interesting.
Mm.
I think we have time for one more question. If nobody raises their hand, I had another question about the species you just mentioned with Olwen, Aloe Bali, because you also mentioned that it's quite toxic, right? And I've seen reports of the species actually being mistakenly used for aloe vera and having caused people to die from ingestion. So I was wondering why this aloe is still so popular, despite being so heavily toxic, especially in beer brewing.
There's no any report for any death resulting from the consumption of the beer from Bali, aloe Bali.
Okay.
But I think they reduce the toxic compound by the way they prepare it.
Mm-hmm.
Yeah.
How do they?
I think they have to. I don't know exactly, but they have to soak it for some time before they mix it with other sugarcane, and other local materials for beer brewing. So they have to soak it first.
Okay. Okay. That's very interesting. Okay, thanks, Siri. That's all we have time for your presentation. But thank you very much. Great job.
Thank you.
And then we move on to our second speaker. So let me just share my slides again. Okay, so our second speaker is from yet another continent. Mehrdad is one of the Plant.ID students from today, and he is from Iran. He studied computer sciences first in Isfahan and then in Tehran. But nowadays he lives in the beautiful Dutch city of Leiden, where he is doing his PhD at Naturalis Biodiversity Center. And he's using his computer skills to make automated identification methods for wood products in illegal trade, such as for violin bows. So please give a warm welcome to Mehrdad. And once I stop sharing my screen, you can go ahead with your presentation.
Thank you, Yannick. Let me just share my screen. Do you see my presentation?
Yes, very clear. Thanks.
Okay, great. So thank you very much for a nice presentation, Yannick, and good afternoon, everybody. In this talk, I'll be talking about my PhD project, which is actually developing an innovative tools for monitoring illegal hardwood timber trade. But before anything, let's just quickly see why illegal trade matters. Well, it's being said that about 10 to 30% of the timber trade is from illegally harvested materials. And it's actually in tropical forests, it's even worse. So the estimation is about 30 to 90%, which is a lot. And the illegal timber commerce market has an estimated annual value of something about $30 to $160 billion. And if you compare to legal market value, which is about like 200 billion, see that it's about 15 to 80% of legal market, which is again huge. And these are just some points that I want to bring to your attention, but actually there are much more. We all know about deforestation, climate change, all the environmental and economical effects. So that's why it is important. But in this project, actually, I'm focusing on just Diospyros species or ebony woods. And Diospyros is actually the largest genus in the medium-sized pantropical family, Ebenaceae. And the genus Diospyros comprises around 800 species of trees and shrubs. There are basically two main groups of species in Diospyros, one called persimmons, and they are very well known for their fruits. And one of like a prime example of these species are kaki, Diospyros kaki with its kaki fruits. You may have seen it in supermarkets. The other group of Diospyros species are those that actually produce ebony woods, and they are valuable for their timbers, not for their fruits. And why are the ebony woods important? Well, as I said, the hardwood of some Diospyros species are colored black, as you see in the picture in the bottom section, and they called ebony or ebony woods. They are extremely hard and are used for making various parts of string instruments, such as fingerboards, tuning pegs, chin rests, etc., in violin, viola, cello, or double bass. And this is the main use case of ebony wood these days, but you may find some antique ebony sculpture or even furniture, but they are not the case anymore. So they are mainly used in mostly musical instruments. And which ebony woods are actually protected? Which one we want to emphasize on it? And the answer is Malagasy ebonies, or ebony from Madagascar. The reason being is that after 2009 political crisis in Madagascar, several timbers, including rosewood and ebony woods, were logged illegally from within protected areas due to lack of environmental governance or law enforcement. But because of that, CITES, the Conventional International Trade in Endangered Species, has put all the Diospyros species from Madagascar in Appendix 2 since 2013. So listing under Appendix 2 does not mean that the trade of those species are completely forbidden, but it needs a proper permit. And it means that they need more attention, basically. And if it doesn't happen, they will be extinct, unfortunately. So what we are going to do, what's this project about? The goal of this project is to build a classifier or classifiers to identify sample of ebony woods based on wood anatomical features and chemical profiling methods to distinguish illegally versus legally logged. So in this case means to detecting samples from Madagascar. And as you see in the picture, in the top right box, we have some wood samples from scientific wood collections, and we use them as a reference material for our training set. And in the left box, top left box, we have some musical instruments, and we use those ebony pieces like tuning pegs as the training set. And in the bottom section, you see two identification methods that we use. In the right-hand side, we have wood anatomy. We do imaging of these wood slides. And then by using a computer vision algorithm, we can identify them up to genus or sometimes species level or clade level, basically group of species based on their morphological patterns. And in the bottom left box, we have Dart of MS, which is another identification method. And in this talk, I will be focusing on Dart of MS. I will show the result of this method. So let's see what Dart of MS is. It stands for Direct Analysis in Real Time, Time of Flight, Mass Spectrometry. And basically it's a method for producing chemical profiles from samples. What Dart of MS does is exposing wood samples to a stream of helium ions heated to 450 degrees Celsius, and then recording chemical peak patterns, as you see in the picture. So by comparing peak patterns, something like this, with to a reference database, we can identify an unknown wood sample up to a species level. So this is very important because wood anatomy usually doesn't have enough resolution to do identification up to species level, especially in a homogeneous general like the sparrow. So by combining these two methods, we can increase the resolution, basically. The thing is that the conventional method is more experimental. They're based on trial and error and also needs background knowledge. You need to know how, for instance, set the thresholds and how to select peaks, etc. But the approach that we are using is actually using deep learning, so in which the convolutional neural network can do many things automatically. Well, of course, there are several parameters involved that you have to set and do training. But once you got a trained model, you can just use it over and over without any, let's say, prior knowledge. So basically, less pre-processing involved, it's faster, and also it's more accurate. So for convolutional neural network, we basically need images. And it means that we have to convert these peaks to an image. And the image of these peaks, if we plot them on a heat map, looks like something like this that you see in the picture. So in the left side, we have Thesperus aculeata. And what you're seeing in this chart is basically peaks. So in the x-axis, you see mass charge ratio from 100 to 1000 AMU, atomic mass units. And the colors represent the relative intensities from 0, where dark blue, to 100% yellow. So then they show the relative intensity. And y-axis actually says that the number of samples that you have. So in this case, it's just 1. So that's the reason it says 1. And in the left, sorry, in the right figure, you see Dalbergia baroni, which is not only a different species, but it's also from different genus and also different families. So it's a Dalbergia from Fabaceae and the Diospyros from Evaneaceae. So I just put these two as an example to see how they looks different from one another, and why we can do identification based on these peaks. Before showing the results, I would like to briefly tell you how convolutional neural networks actually work. A convolutional neural network comprises several layers of basically convolution layers. And it means that each layer does a convolution operation, which is like a dot products, and producing weights or just a couple of numbers, which passes through the networks. And at the end, by combining all these values, then we can produce a feature map. And then by using that feature map, we can do classification and say which sample we had at the beginning. So it's like a very, very simple explanation of a convolutional neural network. One very important point that we have to always keep in mind is that to use DART-FMS, we actually need a splinter of wood samples from hardwood and not sapwood. So as you see in the picture, just a given trunk, you have bark in a very outer side and then light area next to bark, which called sapwood. They are basically includes live cells. They carry sugar, water. And then we have pit in the center, and the area between pit and sapwood called hardwood. So they're basically mostly dead cells. So it's very important to have samples from hardwood and not sapwood, and I tell you why in a bit. But yeah, this is something that we should keep in mind. So before showing any results, just a quick overview of what I'm going to show you. So for a given wood samples, we do first a classification to see whether it's a hardwood or sapwood. So for us, hardwood, we need hardwood basically, not sapwood. But for the people who are working with the DNA, they may be interested in sapwood. So that can be also used for them to find a sapwood sample. And then when we find hardwood, then we can do identification on a genus level, see whether it's Dalbergia, for instance, or Diospyros or Abies. These are like genera that are being used to make musical instruments, basically. So I just put them as an example. And let's say we found Diospyros, then we want to see from which area that sample is coming from, whether it's from Southeast Asia or India or Africa or from Madagascar. And the next level to that could be a species identification, but actually up to this level can also produce a very, very valuable result for us. So yeah, that's also a very nice finding. So hardwood versus sapwood. In here, I put two heat maps, one for hardwood in the left and one for sapwood in the right. And as you can see, sapwoods are very, very, very similar to one another. It's very, very hard to do species identification because there's a very low variation there. And that's the main reason why sapwoods are not very useful for a species identification. There are also some other reasons that if you, for instance, collect data from sapwoods with DART-FMS, then you will end up with caramel, peaks, burnt sugar, and then you have to subtract it from the peaks. And yeah, there are just lots of work to do. But as you see in hardwood, there are more variation and makes it more suitable for species identification. But basically, that's the main result. But that's the main reason we need it. And by the way, all these data that I'm presenting here is from a forest database. A big shout out to our colleague, Dr. Ed Espinoza from Fish and Wildlife Forensic Lab in Ashland, Oregon. He and his colleague did an amazing job to compile these data sets, and I'm very honored to collaborate with them. So all these data is coming from that database. So Diospyros versus Dalbergia. Next, when we found the hardwood, then we want to see which genera is that sample from. So I put here a heat map of around 800 Diospyros species against a heat map of about 1,000 Dalbergia species. And again, as you can see, they are very different. So in the Diospyros, we have a concentration of high peaks around 100, between 100 and 200. But in Dalbergia, there's actually nothing going on, especially around 150, but the concentration is more around 400 and 500. And the peaks are relatively evenly distributed than Diospyros. So it's not very challenging for a convolutional neural network to distinguish between Dalbergia and Diospyros. It can be done up to 98, 99% of accuracy. And it's actually the first step of doing identification. So it's a genus level identification. After that, the geographical distribution is also very useful and informative for us. What you're seeing here is the distribution of Diospyros species that are used for to train this classifier. By using a T-student neighbor embedding algorithm, you can produce these sort of scatter plot, and you can check whether your CNN worked well or not, whether the feature map that is produced is good enough or not. So as you see here, there are five main cloud of data here. So the reds are African samples, the purple one are Southeast Asia, green ones are Indian samples. The blue ones, that's actually very interesting for us, is Malagasy samples or Madagascar and Mauritius sample, and the American samples are just far away. But as you see around this area, it's a little bit noisy, and there are lots of samples of sharing in this area, so it makes it challenging especially for the species that are falling in this area. But generally speaking, we can expect about like 70% of confidence to distinguish different Diospyros species based on their region. So let me show you some real world results of musical instrument pieces that I analyzed. And this is just a general heat map of those samples. And as you see, the general patterns very look like the Diospyros, but we also have some Dalbergia patterns here, which is in line with what we found. So a total of 137 musical instrument pieces were analyzed. And again, a big shout out to Ulrike Wiebel, Renesel, and other violin, cello, double bass bow makers for their contribution. Basically, 1/3 of the musical instrument pieces that I analyzed are donated by Ulrike. 1/3 of them are donated by other NGV and non-NGV members, and the rest are just being bought from web shops to have a relatively diverse samples. So from these 137 samples, 7 of them are from Dalbergia species, mainly Dalbergia melanoxylon, and the rest are Diospyros species. And from these 130 Diospyros species, we have 13 African samples, about 115 Indian and Southeast Asian samples. So these regions are relatively close to one another. And the two main samples of these regions are Ebenum from India and Celebica from Southeast Asia. So they also share lots of characteristics. They're also a little bit challenging to distinguish them, but at least we can say whether they're from Africa or Southeast Asia, Indonesia, or Madagascar. I could find 2 Malagasy samples there, but again, these the total accuracy of these classification is about 70%. So there might be also some errors there, and also some Malagasy samples based on Dartophamus characteristics. They also shared some characteristics with some African and again Indian or Southeast Asian samples. So that some of them are also very challenging. But again, I just want to show you how these pipeline basically work, and yeah, it looks promising. But of course, we can improve it. I'm working on it to improve the algorithm to work with the limited sample size, and also try to increase the number of samples if I can. And also I'm working to combine it with wood anatomical microscopic images to have a better identification tools. So for some challenging species in Dartophamus, we can use wood anatomy and see whether they are if they belongs to same species, or they are 2 or 3 different species. And last but not least, we're also working on C14 carbon dating as a dating method to estimate the date of harvesting of a given sample. So at least we can see whether a musical instrument, even if that's from Madagascar, whether it's an antique sample or it's more recently harvested sample. So that's also very important. And at the end, I would like to thank all the people that helped me, my supervisors, Barbara Herfandil, Frederick Lenz, and Fons Verbeek. Also our colleagues, Ed Spinoza and Katie Lancaster, to compiling and helping me for Dartophamus sections. Ulrike Wiebel, also students who helped me to produce all these results. And also thanks for your attention. If you have any question, I'll be happy to address them.
Thank you, Mehrdad. Great presentation, and I like that you're actually presenting it from within a microscope slide.
Yeah, just forgot to mention, if you're wondering what is in my background, it's actually Diospyros perrieri from Madagascar in transverse section. So yeah.
That's really cool. Really cool.
Thank you.
Yeah, thanks for making a complicated topic a bit more accessible. We are running a little bit late, so I'm just gonna do one question for this session, but if anyone else has any questions, please feel free to drop them in the chat, and Mehrdad can answer them later. I see Pete Gasson is here and he has unmuted himself. Do you want to ask a question, Pete? Oh, we can't hear you. I think your microphone might not be working. No, still can't hear you, sorry. Sorry, Pete, we can't hear you. I think your microphone's not working. Yes, now we can hear you, yes.
Do you have many authentic samples of Diospyros from Madagascar? Because that's a problem we have. We've got hardly any at Kew.
Yeah, basically that's also the same challenge I have. The number of samples that I have from Madagascar is very limited. It's actually about 80, 90 specimens. Well, some others are just samples, so I cannot use them. So yes, this is basically the main challenge. but I try to at least improve the algorithm in a way that it can deal with the limited samples. But yeah, I agree. This is just a main challenge that we have.
Yeah, because I suspect that the anatomy is going to be a bit unhelpful. I think most of them look very similar to each other.
Yeah, I agree. So hopefully Dartov MS has a higher resolution in that regard. But again, yeah, the sample size is the major challenge. So maybe if we can combine those methods, it can be improved.
We had our DART machine delivered this morning. It hasn't been plugged in yet, but fairly soon we'll have a similar machine to the one that you're working with.
Oh, that's great. That's a great news. So hopefully can collaborate in the future.
Well, we'll be in a better position to do that, having the toy with us now, rather than having to rely on Ashland and other places.
Okay, it's great.
Looks like you're forging good collaboration here. That's exciting.
Yeah, let's hope so. Yeah.
Yeah.
Well, I have heard from Vicky Beckers. I think she was asking us for some samples, but I'm afraid we haven't got very many. So I don't think we've been very helpful there. You certainly have a lot more samples from Madagascar than we do.
Well, yeah, but these are samples I mainly collected from Terfurum wood collections. So yeah.
Well, if you don't know, do you know Victor de Klerk?
I've heard of him, but I haven't met him in person.
Right, well, he's worked with Hans Beekman, and he's starting working at Kew on Tuesday, and he physically arrives on the 14th. So I think some good collaborations are in the offing there, because he knows how to use these machines. I don't.
All right.
Okay, so yeah, that's great.
Sounds like the two of you need to have a good chat.
Yeah, we do.
Yeah. Okay. Thanks, Pete, for your question, and thanks, Mehrdad, for your presentation.
Great talk. Thank you.
Thank you.
Great. Yeah. So we're having a little break now. We're running a bit into the break, so we're going to make it a bit shorter. But let's try to come back here in 10 minutes, around 3 for UK people, 4:15 for European people. And then Olwen will take over chairing for the session. So grab some coffee and see you in a few minutes. So when I'll just unshare my screen and then you can take over.
Yeah.
Except that I don't have anything to share myself.
Did I not send you the slides?
It's probably easier, Yannick, if you continue to share your screen and I'll just do the talking.
Okay, that's
Is that
fine.
all right?
Yeah, of course. Yeah.
Thank you.
Yeah, no worries.
We'll just give everyone another minute or so to get settled.
Yeah, exactly. We'll see the camera switching on again. A very good talk so far.
Yeah, and it's lovely to see everyone here today.
Yeah. And I see Mehrdad has already started answering some of the questions.
Yeah, he has. Yeah. All right, let's get started.
Yeah, let's do it. Okay, I'll go to the next slide.
Okay, so welcome back, everyone. Thanks so much for joining us this afternoon again. I'm delighted to introduce our second Plant.ID speaker of the day, Maurizio Mascarello, who is in his final year of his PhD at the Botanic Garden in Meise and the Catholic University Leuven in Belgium. And his project is really about developing new molecular markers for African timber identification as well. So Maurizio is originally from Turin in Italy, where he obtained his bachelor's and his master's in environmental biology, then a second master's at University College Dublin, and then on to Munich as a research assistant. So Maurizio certainly fulfills the mobility requirements that our funders require. Anyway, a very warm welcome to Maurizio, and we look forward to hearing all about your research.
I would like to share the screen, but I cannot because there is already the slide.
I think Yannick, if you stop sharing.
Okay, okay, perfect. Okay, now I can share.
Is it working?
Yeah, sorry about that.
No worry.
Great. Thanks so much, Maurizio.
Yeah, can you see the presentation?
We can. Can you make it larger?
Yeah. Okay. Perfect.
There you go. That's
okay.
perfect.
So, good afternoon, everyone. My name is Maurizio Mascarello. I'm a PhD researcher at Meise Botanic Garden and KEW Leuven in Belgium, and I'm also a member of the European Network Plant.ID Molecular Identification of Plants. Today we'll give a presentation about the use of genoscheming to find novel plastic marker for the identification of illegally logged African trees. So the main project partners in my project are Professor Harris Metz, who is the promoter of my PhD and the scientific director of Naturalis Biodiversity Center, and Dr. Steven Janssens, the principal investigator of the project and senior scientist at Meise Botanic Garden. So in the mid-20th century, the demand for timber by China and Western countries drastically increased. This growth was supported by the enhancement of harvesting, processing, and transportation technologies. And as a result, the increased pressure by logging operation caused a massive deforestation and loss of huge forest area, which also negative consequence of the global conservation in the tropics. This image showed worldwide timber trade. The main world importer is China. However, we can see from this image that European Union is the main importer of timber from the Congo Basin. So despite the restrict logging concession and improved forest regulation, up to 90% of the annual timber volume is estimated to be obtained through illegal logging activities in the Congo Basin. Despite the use of strategy to track logs from the harvest to the final destination, criminal cartels can efficiently use strategy to overcome the control by both local police and international authorities. These strategies include logging beyond concession, falsification of export permits, and the mixing of illegally logged timber with timber obtained via official procedure. So the first forensic technique used for species identification is the microscopical analysis of wood anatomical characters. This was also supported by the DNA barcoding, which is the use of a genetic marker for species discrimination. However, none of these techniques allow the universal identification of 3 species. In this slide, I show an overview of the conventional DNA barcodes used for species identification. The first one is the cytochrome oxidase 1 in the mitochondrial, which is an excellent marker to discriminate animal species, but it exhibits a variability which is too low in the plant kingdom. So there were developed markers at the chloroplast level, for example, the coding regions of the MATK and RBCL, but these markers are not universal because they exhibit low discrimination or no discrimination power at all in some group of plant species. So furthermore, were developed markers for the amplification of the ITSs, ITS1 and ITS2, which offered a higher discrimination power, but these have the limitation that have paralleloscopy in cells, and they may bring to detect fungal contamination because the conventional primers to amplify ITSs are similar for the plant kingdom and the fungal kingdom. For this reason, the development of novel molecular technologies is essential to enhance the identification of 3 species in order to mitigate illegal logging activities in the tropics. In my project, I opted for the genome skimming method, which consists of the sequencing of the total genomic DNA at low nuclear genome coverage. This techniques is ideal for the reconstruction of high copy plant organelle genomes, is suitable for the analysis of degraded DNAs, and is also useful for the identification of novel genetic markers. As a main target, we opt for the chloroplast genome because it was found to exhibit a conserved gene order and a lower mutation rate compared to the nuclear genome. It was found to be excellent for species identification and for phylogeny resolution. So the purpose of my project is indeed to use advanced molecular techniques to stop illegal logging activities of African trees in support of the European forestry law and regulations. So firstly, I selected species from tropical Africa, especially focusing on Central and West tropical Africa, species of economic importance for the international timber trade. And I'm focusing especially on species which critical conservation status. Then I perform DNA extraction from herbarium specimen and silica dry samples obtained from various botanical collections. Then I perform the DNA library preparation, focusing on the optimization of the protocol to process degraded plant material. The protocol consists on a main enzymatic fragmentation of the total genomic DNA, followed by the ligation of the resulting fragments to a universal adapter for Illumina sequencing. Then we perform the site selection of the fragments suitable for the target sequencing strategy. We perform, of course, a PCR enrichment to increase the sequencing success. And finally, a cleanup and quality control to maximize the reliability of the quality of our sequence. We perform the quality control by capillarity electrophoresis using a fragment analyzer, which allow not only to get a reliable concentration, but also to evaluate the fragment size distribution of our samples. Then we send our sample for high throughput sequencing. We opt for the pair hand 150 base pair strategy using Illumina high-seq technologies. Then we perform the bioinformatics analysis on the Illumina sequencing reads. First of all, we perform the de novo assembly of the chloroplast genomes, followed by the genome annotation and the multiple alignments of the species in this study. This in order to search for highly variable regions in order to select single nucleotide polymorphism for species identification. So for genome assembly, I choose the pipeline GetOrganelle, which is a very user-friendly pipeline for the de novo assembly of chloroplast genomes using genome skimming data. For the genome annotation, I use the online pipeline CP-GABAS2, which I also recommend for this purpose. So this image shows an example of a complete chloroplast genome and its respective annotation. And from the comparative genomic analysis, I can confirm that the conventional plastic markers do not always discriminate among African timber species. These markers include the coding regions of the MATK and RBCL genes, the TRNL intron, and the intergenic spaces TRNL, TRNF, and TRNH PSBA. We found gene losses occurring in specific clades and families. For example, the INF8 gene we confirm that was lost in Rositz clade, and that the RPL22 gene was lost in Fabaceae. Then we found we could confirm that in the subfamily Papilionidae of the family Fabaceae, there are several species exhibiting a 50 kb sequencing version in the long single copy regions. And we found particularly that the genera Milletia and Pericopsis present intrinsic sequencing versions of 6 and 2 kilobases respectively. Then we identified highly variable regions for the identification of a high number of species. At the level of coding regions, the coding regions of the NDHF and the YCF1 present high variability in terms of high number of polymorphic variations in several species. In this image, this is an image of the polymorphic variability in Pauteria species, and it shows indeed that while the MATK and RBCL exhibit a very limited variability, the NDHF and the YCF1 present a high polymorphic rate. So then we found several AT-rich microsatellites region which are not suitable for species identification. Among these regions, one of these regions is the conventional marker TRNH-PSBA, which exhibit a high AT content, and this explains why this region was found to have to offer bad sequencing results from Sanger sequencing data. This is indeed an example of a intergenic spacer in Afzelia, which exhibit two AT-rich regions with a very high variability, which is not reliable because it may lead to sequencing mistakes, and these areas are abundant in families in which the total AT content is high, like Fabaceae, for example. So, from the results of genomic barcoding from genome skimming, the purpose, I'm now working on developing a novel genetic marker for DNA barcoding. So the purpose of this first part of my project is to go from genome to barcodes. This because the high throughput sequencing is still a long and expensive process, so that many institutions, especially in the developing countries, cannot afford high throughput sequencing technologies. On the contrary, DNA barcoding can be carried out by the majority of scientific and forensic institutions. Therefore, summarizing, the development of short species-specific genetic markers, alias mini barcodes, is essential for species identification, for example, of timber species under the wildlife trade of valuable timber. So the approach I'm now working is, first of all, always start from the genome skimming of a group of major commercial timber species, especially focusing on obtaining the plastome and the nuclear ribosomal genome of multiple individuals from the target species. This in order indeed to de novo assembly the chloroplast genome and the nuclear ribosomal genome for this species, in order to evaluate the intraspecific variation in both plastin genes and ITSs. This in order to confirm single nucleotide polymorphism reliable for species identification, and in order later to develop genetic marker and test them through PCR amplification on both reference specimen and commercial wood samples. This in order to increase the plant barcode database with reliable marker for the forensic analysis of exploited African timber species. Thanks for your attention.
A round of applause, please. Thank you very much, Maurizio. That was fascinating. We are running a few minutes behind, but I would like to give you the opportunity to answer one or two questions. So I'm just going to have a quick look to see who's got hands up. Wu, would you like to come on microphone, please?
Yes, Maurizio, thank you very much for the interesting talk. I was just wondering, because you have the genome skimming data and you assemble the plastome, except for the ITS, have you assembled other genes from the nuclear genome, which, if you did, how does that gene works? How does those loci works?
So I'm currently assembling the complete nuclear ribosomal genome. At the moment, I'm only analyzing ITS, but of course with a proper pipeline for the pipeline get organelles work mainly for the chloroplast, but is also good for the nuclear ribosomal genome because it exhibits a very high coverage. And so far, yes, indeed, for with this strategy, you are also able to obtain other genes in the nuclear ribosomal genome. So this is possible through this through the genome skimming technique, because the nuclear ribosomal genome exhibit a very high coverage.
Yes, that's what I'm thinking, because I'm using the nuclear genome trying to tell the species apart. I was wondering how your data is looking like. It will be interesting to see your data comparing to the plastom genomes and how does it work. Thank you very much.
What I'm doing. Thanks.
Thank you. So thank you so much, Maurizio. There are other questions. There's at least one other question for you in the chat, which you can respond to, but we should press ahead with the program. So moving to our final speaker of the day, I'm delighted to introduce Dr. Carly Cowell here from the Royal Botanic Gardens at Kew, who's a senior science officer leading Kew's policy team. Carly started her career in horticulture in her home country, South Africa, and went on to obtain degrees in ecological restoration and biological conservation management. And she also obtained her PhD degree from the University of Cape Town, working on the role of past and present botanical collections in conservation practice. She's published on topics such as ecological restoration, conservation policy, plant blindness, biocultural heritage, and illegal wildlife trade. She's a member of the IUCN Mediterranean Ecosystems Working Group and the Society for Ecological Restoration. And as such, she's involved in some important international legislation and decision-making. And so we're very grateful to have Carly here this afternoon to provide that broader context for what we've been discussing and hearing about in the other talks of plant uses, threats, the challenges of identification and their application for trade monitoring. So many thanks, Carly. Over to you.
Great. Thanks, Olwen, and thanks, everyone. I hope not to bore you. The talks so far have been really fascinating and now slightly different. I'm just going to share my screen here.
Great, something's happening.
Something, let's hope.
Okay.
There we go.
Got it.
Okay.
Great, thanks, Carly.
Sure. So this afternoon, I'm just going to run through the CITES regulations and the importance of the work that you're all doing in tracing illegal trade in plants. So I'll give a brief background of what CITES is and the aims of the convention, and previous speakers have already alluded to CITES. I'll explain the species listings and what trade we see of CITES species, and then I'll run through some challenges of enforcing it and the need for DNA and technology to help us conserve more species. So why do we conserve wildlife? Well, those of us who work in conservation, we often forget that it's not at the forefront of many people's minds or a priority. And it's always good to consider why people may want to conserve a species, as it differs across countries and cultures. And for instance, there's the basic utilitarian benefit for consumption or non-consumptive use, ecosystem services, particularly around climate change. There's a bequeath value for the future generations, for our children. There's an altruistic view of if other people enjoy it, then we should be conserving it for them. And then there's just the pure existence value in that wildlife has the same right to exist as we do. And we deal with this in CITES at multiple levels across country scale and then through individuals. So all of these factors came to play back in 1973 when the first parties to CITES met in Washington, DC. and the Convention on International Trade in Endangered Species came into being. It was only officially ratified in 1975, and it was the first global convention for the protection of species. It's good to remember that at that time, there was no internet and international travel was very limited. I'm not sure if it was as COVID limited as we have experienced recently, but enforcing CITES at that point was slightly easier than what we are experiencing today. So why was it created? And apologies for the leopard in the screen, but it's a fantastic shot from my country. I had to put it in there for South Africa. But wild species were becoming increasingly threatened by trade at that point. It was new. Now we know that trade can threaten species to the point of extinction. But back in 1973, this was a new concept. And it was decided that control of trade was required with international cooperation. So CITES has its mission, its vision statement, which is to ensure that no species of wild fauna or flora becomes or remains subject to unsustainable exploitation through international trade. And CITES has these three objectives to achieve this: under legality, where specimens are legally obtained; sustainability, where wildlife trade is not detrimental to the survival of the species; and traceability, that trade can be traced from source to final destination. And in all three of these objectives, the work that this group is doing and our presenters who've presented this afternoon fits into all three of these objectives. So just some stats on CITES. There's currently 183 parties who've signed up to the convention. Those are the ones in green. There's a few yet to sign up, but majority we can say it's a global convention. It follows the same as other international conventions that you may have experienced with the Conference of Parties, which meets every 3 years, which is the major decision-making body where they consider proposals to amend appendices where the species are listed. And the proposals are assessed against very strict criteria by each country. And if any decisions are made, it has to be voted on with a 2/3 majority in the actual sitting of COP. It also adopts decisions and resolutions for the administration and operation of CITES, and then agrees on a budget. So how CITES works in the years between the Conference of the Parties is that it is advised by a standing committee of select members from the parties and observers, and then a scientific committee for animals and one for plants. It's administrated by the UN Environment, and our current Secretary General is Yvonne Higuero. So CITES works on a permit system, which I'm sure many of you have experienced in your research requirements, but it regulates the international trade of wild specimens through a permit system. So there's import, export, re-export, and certificates of use. Now, certain countries, or any country, may decide to implement stricter domestic measures as they're known. And for example, the UK and the EU, we have wildlife trade regulations, which are slightly stricter measures than CITES enforces. So for instance, for the UK, we require export permits, import permits, and re-export permits for everything coming and going under CITES to and from the UK. Some countries just require export permits if they are a range state. So for instance, the timber examples which we've seen earlier, the Congo just require export permits to a country of destination. That country may not require import permits if it's not under a stricter domestic measure. So as we have heard, there are different appendices on CITES, and there's 3, Appendix 1, 2, and 3. Appendix 1 is for species threatened with extinction, which are or may be affected by trade. International commercial trade in wild taken specimens is generally prohibited. It is allowed when species are captive bred or artificially propagated in the case of plants. And currently on the list, there's about 687 animals and 395 plant species. This makes up only 3% of the current listings on CITES. So examples of Appendix 1 plants include Ariocarpus cacti from Mexico, Pachypodiums from Madagascar, and cycads from South Africa. Appendix 2 are species that are not necessarily threatened with extinction, but for which trade does need to be controlled to avoid their becoming threatened with extinction or extinct, and species that may resemble those species that are already listed on Appendix 2, and these are referred to as lookalike species. International trade is permitted but highly regulated, and on this appendix there are about 32,364 plant species, making up about 97% of all the listings. Examples include the Delbergias, which we've heard about, and the aloes, as well as common snowdrops, Galanthus species, and cyclamen, which you may have flowering in your garden in the north here in springtime. Then we have Appendix 3. So these are species for which a particular country is asking for help, and international trade is allowed and regulated, but not as strict as Appendix 2. There's only 9 plant species on Appendix 3, and of those, some common examples are Quercus mongolica, a Russian oak, and the Coco de Mer, the Seychelles palm nut. Now, there's often a misconception that CITES bans all trade. And if you look at this slide, you can see that of the 37,000 or so species that are listed, only 3% are currently listed on Appendix 1, which bans commercial trade. And as I said, but of those 3%, if they are indeed artificially propagated, then trade is allowed. So trade is more regulated than banned under CITES. And then of those 97% that we do have in CITES, we don't see everything that is listed on CITES in actual trade. We did a recent analysis of the 37,000 plus species, and we found that 4% are commonly traded, and an additional 1% can be considered as highly traded. That equates to about 1,800 plant species that we see in CITES transactions on the CITES trade database. Now, the way CITES is broken down and managed in each country is that each party must designate one management authority and one scientific authority. Now, the management authority role is to issue the permits, do many operational activities such as represent the party at meetings, present proposals, communicate with the secretariat, training, and report for illegal and legal trade of CITES. The scientific authority role, which is what I do at KEW for the UK for plant species, is to advise the managing authority that trade in any species is not going to be detrimental to its survival in the wild. And we do this through a non-detriment finding. Now, this looks specifically at the impact of harvest and trade on the survival of the species. We often require information such as the conservation status, population size and structure, biological characteristics, harvest regimes, harvest techniques, and management. And there's much more information that is required to make a positive NDF, as it's known, and then advise the managing authority that the trade can take place. We also do determination of whether a recipient of a live specimen can house and care for that specimen. Generally, that's for animals. We also monitor all export permits and import permits. As the UK, we are quite a consumer country, not a producer. And then we often conduct research on particular CITES species of concern. Then there's 2 more elements to managing CITES and implementing it, and that is customs and the police. So the role of customs is to look at the presentation of the permits at the border, on the ground. They need to be endorsed by a customs official who often needs to inspect a shipment and identify a shipment. And then they do play a key role in ensuring that any red flags where shipments are coming in without the correct paperwork, incorrect species names are being used, that they feed through to us in the scientific authority and the managing authority. And we can look into this, engage with countries, or conduct research. As I mentioned, the police are also important role players, and their primary responsibility is enforcing domestic control and not international control. But from a domestic point of view, they often enforce wildlife regulations inside a country. They share intelligence with customs. They often seize specimens and have samples that we can use to identify and for research. And they often act as a deterrent to any potential wrongdoers at that time. So in CITES, what is actually controlled? So certain species are covered, but when we say control, so it's any animal or plant, alive or dead, or any recognizable parts or derivatives of plants and animal species. And now we get to the fun bit of actually implementing CITES. The term readily recognizable, what does that actually mean? Well, you can either recognize it outright as a CITES specimen. We could all recognize a rhino if someone walked it through an airport. We'd also recognize a full aloe in full bloom in a pot. However, a lot of the things that we see in trade aren't the actual raw product or live animal or plant. So we often have to look at the label or the packaging, accompanying documentation that says it has CITES in it. So for animals, there's things that contain tiger and bear bile, which sometimes it is on the packaging and sometimes it's not. Plants, we have cactus in medicines. We have Hoodia for slimming tablets. We have Dendrobium orchids in all sorts of formats. And if it's listed, it's fine, but what happens when it's not on the packaging and we suspect it could be? So the kind of things that we are seeing in traded plant products, this is everyday items that consumers take for granted that may contain endangered species. So salep, for instance, on the left of the screen, the middle coffee cup image, refers to the dried tubers of terrestrial orchids ground up into a flour, which is often made into a hot drink, an ice cream even, and sometimes used just to make powdered milkshakes. Then we have cactus extract and we have orchid extract, which are used in cosmetics, but are they Appendix 1 or 2? What permits do we actually need to apply? Agar oil, which is on the top right of the screen, is more expensive than gold by weight. It's used in perfumes and incense. And then in the middle, we've got ginseng. We all know it's a supplement, but did you actually know that it was CITES listed and how to identify this? So the graph on the screen just shows some illegal seizures from the European Union in 2015. And the interesting thing here is that the majority of the illegal seizures were for medicinals, which was 63% of those seizures. And of that 63%, 59% was plant-derived. But that's all we knew. We didn't know which plants. We couldn't really follow many prosecutions because of the fact that we need to actually identify what species, what plant, get really down to the basics. Sometimes the traders make it easy and it is on the box. Most times it's not. And the difficulty is that we are seeing a rising demand and the risk for traffickers to do this is minimal because of the high returns. There's also inadequate awareness. People are naive and don't realize that all orchids are listed on CITES. There's resource constraints. Not all customs officers know what plants really look like in trade. They're so busy looking for illegal contraband and other things that they don't spot the CITES species coming through. And there's often low penalties and prosecutions for culprits who are caught. And this is a judicial system that needs to be educated on the value of plants and the threats to them in the wild. Now, we've got a lack of technological equipment and we have a lack of forensic tools and laboratories globally to try and enforce this. And this is where we really need to focus as a research community. So the current tools that we are seeing that have been developed and are working well is X-ray technology for ivory and animal bones. And on the screen, Border Force have provided me with some pictures to show you of what these things look like to the naked eye and then under X-ray. And the green is ivory. It highlights as green in the X-ray. There's also DNA tests for animal tissues, and there's quite quick turnarounds for this kind of thing, particularly for rhinos and when it comes to bear bile and things like that. But for plants, major work is needed. First, we need technology to find the plant material in the packages and the luggage. And with this, we need simple machinery. And then we also need to identify the species once they're actually found in the shipments. So this example here are seizures that were from a joint working operation with UK Plant Health and the Inspectorate for Customs of both ports and international mail depots. And it was a countrywide, a global operation that took place over a period of months. And in the UK, we seized some seeds that were detected in a mail parcel from Mozambique with no CITES documentation. These are cycads, and until the plants are grown or the seeds germinate, we don't have a better way of telling what they are in order to actually put them through into a court procedure as an encephalatus species. And then we can actually look at what is this threat to that particular species and actually make a case for getting a strict sentence on this. We also have the problem of identifying wild versus artificially propagated specimens. So CITES is about wild taken specimens, and generally trade is easier, and I wouldn't say freely allowed for artificial propagated specimens, but then we have to tell the difference because a lot of bad guys want to try and launder species as artificially propagated when they've been taken out the wild. So it's extremely difficult to do this. And I do believe that DNA and isotope analysis could help here. And from what I've heard from our speakers before me, the signs are good. The research is happening for timber at least, but we certainly need to do more on other CITES listed species. And then there's the parts and derivatives, which we have to deal with, which is a more technical issue. It's really difficult, and we're seeing more of this trade than ever before in this history of CITES. More supplements, we are taking more vitamins in general as people, and supplements and all sorts of things. And of course, people are using plant-derived plant-derived resources, and many are CITES listed. So we need to critically look at these because firstly, for human health and safety, as mentioned earlier, there are some aloes that are toxic, yet could be used in place of other aloes which are non-toxic. And then for the survival of the plant species itself. So in summary, that CITES is an international agreement between governments that ensures that no species of wild fauna or flora is unsustainably exploited for international trade. The convention establishes international legal framework and a common procedural mechanism for the prevention of international trade of endangered species and effective regulation for others. So it really is coming together with systems globally in order to do this. It regulates the international trade through its appendices with a system of permits and certificates, and these are issued when certain conditions are met. And it is a powerful tool in achieving consistent international regulation of trade and for sustainable use. And there have been really good examples of species that have come back from the brink because trade has been regulated and people have sat up and realized. However, we need more technological support to identify species and particularly parts and derivatives in trade. And this kind of research takes a long time. So we need to start now because the species are being lost from the wild and their populations are declining now. We need to start our research to get those answers and those practical tools to guys who can implement it like customs and police and actually start saving these species. Oh, thank you. That is me.
Thank you so much, Carly. That was absolutely fascinating. I know I had several questions. That was a really exactly the context that I was hoping you would provide us for as the backdrop of all this other more detailed work that we're doing. So who would, I know that it is already 4 p.m. or 5 p.m. where you are in Europe, But I would like to give the audience the opportunity to ask Carly at least 1 or 2 questions. I'm sure you all have some. So I'm just changing the view here. You can come on mic or raise your hand. No? Great. Hugo.
Hi, Carly. Thank you very much for this very interesting talk. I was wondering about the new proposal for products that contain orchids. How you look at, how CITES should regulate products that are made with CITES listed species, but where we know that they are cultivated.
So yeah, there has been actually quite a bit of research done by the Swiss Scientific Authority looking at this point of orchids being used in cosmetics, and it goes back to that traceability. If you can trace that supply chain, and this applies to all CITES species from origin, and show that, well, perhaps species were taken from the wild, they had to all originate in the wild at some point, but they were done correctly with permits and sustainably, and then grown artificially and maybe propagated via tissue culture, and have that documented. And tissue culture laboratories do document, the species that they go through, and the different media that they use and how long, and that kind of evidence shows that something has been produced, artificially for quite a long time. And then it goes through to the manufacturing, and you can see that those plants were, sort of sent to the manufacturer, and nothing else was received at that point from the wild. And the product itself can then be certified to have artificially propagated species, sustainably sourced, and not a threat to anything in the wild. And then I believe if you can get that certification for manufacturers and producers, then it can be exempted from CITES controls. And there are quite a few species that are already exempt with these checks and balances in place.
Thank you.
Thanks, Carly. I'm aware that we have run over. Does anyone have a quick question they would like to ask? No. Carly, it just remains for me to say thank you so much for joining us this afternoon. And before we all leave the seminar, we'd like to take a photograph, or rather a screen grab of the audience. If you're not comfortable having your photo taken, please turn your cameras off, but otherwise turn them on and join us for a photograph before we say goodbye.
Getting ready here right now.
Smile, everyone.
Just a second. It's
the Plant.ID.
There we go. I got it. Thank you.
Great. Thanks so much, Marcella.
Thank you.
And thank you all for joining us this afternoon. It's been really good to see everyone. We look forward to seeing you at the last of these seminars on the 24th of June. So we will be obviously advertising that closer to the time, but put it in your calendars. Molecular monitoring, plant product quality, and the environment. We'll see you there. Bye, everyone. Thank you.
Bye.
Bye, everyone. Thanks for joining.
Bye-bye.
Thanks.
Bye.
Bye.
Bye.